और Botany? Good evening, one and all. Hello.
Good evening, sir.
Today topic is respiration. Respiration, I think it is in third, third or no, third block. Right. Third block, unit 7, respiration. So already photosynthesis, second block, I have sent in group. Did you see the video? Hello? Hello?
Uh, no, sir. Tomorrow we will see, sir. Or else
Okay,
after the class.
okay. Actually, I have just now I sent it. So see it later. Okay, and complete photosynthesis. In photosynthesis, light reaction and carbon assimilation, C3 cycle and C4 cycle, they are all explained well. So, and this is the connection to the respiration. Let me give brief introduction about photosynthesis. After that, we will continue through respiration stages. So first in photosynthesis, what happened in photosynthesis? You tell me, what is photosynthesis first? What is photosynthesis?
Photosynthesis, sir.
Right, photo means light.
Synthesis means a reaction.
Here the what synthesized during photosynthesis. Photo means light. Synthesis means food material synthesis. To prepare food material, here the requirement of to photosynthesis, the chlorophyll pigment, carbon dioxide, water molecule, carbon dioxide and water molecule, chlorophyll pigment in presence of light to prepare the food material. What is the food material? The carbohydrate. Where the carbohydrate prepared? Hello? Hello?
Yes, sir.
Where the carbohydrate formed? Where photosynthesis takes place?
Chlorophyll, sir.
Chloro,
Chloroplast.
chloroplast. Actually, this photosynthesis two type. One is light reaction, or one is the dark reaction. In light reaction, only the ATP, NADPH energy formed. I will explain clear. Actually, I have explained clearly in my video that I have sent to you in group. Uh, and after formation, this energy, NADPH, ATP energy, this ATP, NADPH energy utilized for the synthesis of food material, that is carbohydrate. So anyway, the carbohydrate formed in stroma of the chloroplast. After that, carbohydrates diffused into the cytosol. The carbohydrate may be in the form of glucose, in the form of fructose, or in the form of sucrose. Any form diffused into cytosol, right? Okay. After diffused into cytosol, now the respiration start. So what is the substrate to the respiration? Let me take white paste. Just wait. Anyway, actually, what happened here? For example, Chloroplast. These are the chloroplast thylakoids. For example, this is the cell. For example, this is the cell. Right. In cell only I have taken and the cell, this is chloroplast and somewhere there the mitochondria. Are you able to see this diagram? Hello? Hello? Mariam? Hello?
Yes, sir.
Diagram visible or not?
Yes, sir. Yes, sir.
Right. So during photosynthesis, what happened? First, light reaction takes place in this thylakoid membrane. Thylakoid membrane. Tejaswini, get it out?
Sir, clear, sir.
Right. Here, this is thylakoid membrane. Here, the light reaction takes place as a result formation of ATP, NADPH. After formation ATP, NADPH, this energy, that means this ATP, NADPH energy utilized for the next reaction. What is the next reaction after light? Dark reaction. In dark reaction, what happened actually? In carbon dioxide assimilation takes place. In carbon dioxide assimilation from the atmosphere, carbon dioxide received, carbon dioxide received, and with presence of water and chlorophyll pigment by utilizing ATP and ATPH energy, convert into carbohydrates, for example, glucose. Now where it is, now it is stroma region. This is thylakoid membrane, but the rest of the photosynthesis takes place in stroma. This is this empty place stroma region. In stroma region, this carbohydrate formed. Later, this carbohydrate diffused into cytosol. Now, for example, this is the cell. This box is the cell. In what are present inside the cell? Cell, all cell organelles are present. So one of the cell are chloroplast. Here the mitochondria. Now between the cell organelle place, this empty place, this is called cytosol. This is called cytosol. Now in cytosol, glucose diffused. Now this glucose act as a substrate. Now this glucose act as a substrate. Now the substrate for the respiration. Without glucose, there is no respiration. Now this glucose enter into respiration through the three stages. What are the three stages mainly? Tell me the three stages. First of all, the first reaction, glycolysis. Glycolysis. After glycolysis, end product is pyruvate. I will explain later clearly. After formation of pyruvate or pyruvic acid, now the pyruvate or pyruvic acid enter into the mitochondria. The rest of the respiration stage takes place in mitochondria. And in mitochondria, the Krebs cycle takes place. That is the second stage. Krebs takes place. I will explain later the Krebs also. And after that, electron transport system formed inside the mitochondrial membrane, in crystal membrane. In crystal membrane, electron transport system formed. The finally, as a result, ATP energy formed in mitochondria. ATP energy, already told you, already we know, these are the powerhouses. ATP synthesized in the mitochondria. So this is the cycle. So first respiration, so first photosynthesis. In photosynthesis, end product glucose formed. The glucose diffused cytosol, where the respiration start in the form of glycolysis. After that, after the end product of glycolysis, pyruvate entered the mitochondria, where the Krebs cycle takes place. And after that, to production of ATP, the electron transport systems occurs. As a result, ATP energy formed. So this is the overall concept of the respiration and photosynthesis. Let's discuss one by one. First, discuss glycolysis. Glycolysis, it is partially oxidation process. The partial oxidation process, end product pyruvate, right? The later, after complete oxidation takes place, ATP energy produced. First, one by one, first let's discuss glycolysis process, right? So in glycolysis, I keep it a slide. Glycolysis is visible to you? Hello? Glycolysis. Hello? Please respond.
Sir, yes, sir.
Right. Glycolysis. So this is the first stage of respiration. Already told you what happened during the respiration, during glycolysis. Pyruvate acid at pyruvic acid formed, right? Let's see the intermediate stages of this glycolysis. First, what is glycolysis? Glycolysis means glucose lysis, lysis into pyruvate. So that is called glycolysis. And where it is occurs, where the glycolysis takes place in which region? Hello? Already told you just now. Glycolysis takes place in cytosol region of the cell, not cytoplasm. Remember, there is difference between cytoplasm and cytosol, including cell organelles, cytoplasm. Without cell organelles, cytosol. That is the difference. Okay, and this another name of the glycolysis Emden-Neurath pathway, because this cycle discovered by Emden Meyer. That's why it's called as Emden-Neurath pathway. Another name is exose diphosphate pathway, HDP, exose diphosphate pathway. So these are the synonym of the glycolysis. Okay, remember these two words: glycolysis, EMP pathway, Emden-Meyer pathway, exose diphosphate pathway. And the another name is, this is the core respiration. This is the core respiration. Then this is the first respiration stages. Right? Is it clear? Hello?
Sir.
Right. So glycolysis substrate is glucose. Already told you the respiration substrate glucose as it first stage glycolysis. So the substrate is glucose. Right? Now discuss the stages of the uh, glycolysis. First one, this is the substrate. Already told you how the glucose formed in plant. Okay. In how it is formed. Glucose formation by, hello, hello,
Yes, sir.
glucose formed by, carbohydrates formed by, how the glucose carbohydrate formed in plants
With the help of sunlight, sir.
with the help of sunlight photosynthesis process, right? So
Yes.
first of all, this glucose phosphorylation reaction takes place. During phosphorylation, what happened? ATP, adenosine triphosphate, convert into ADP. Here the one phosphate, here in triphosphate, one of the phosphate utilized by glucose. So this reaction called as phosphorylation. And for this reaction, One enzyme is utilized, that is exokinase glucosinase. Exokinase or exokinase enzyme utilized for this reaction. So this is called as phosphorylation. As a result, formation glucose as add by another phosphate, it is converted into glucose 6 phosphate. Glucose 6 phosphate. So this is the first reaction. This is the first reaction, right? And second, isomerization takes place. In isomerization, the glucose 6 phosphate converted into fructose 6 phosphate. Glucose converted into fructose. The enzyme is isomerase. Enzyme is isomerase, and this reaction called as isomerization. If the isomerization means actually there is no change in the structure, only change in the groups only. Change in the group. For example, here the carbon, here the carbon, here also carbon, carbon 1, 2, 3, 4, 5, 6. So another one also 1, 2, 3, 4, 5, 6 carbon. For example, here OH group right side. Maybe this first carbon OH group left side. Only the right and left mirror image is like. So this is called a isomerization, but never change the formula. The both glucose is 6, H12O6, and here fructose also same formula, right? So there is no change in glucose and fructose, and this process called as isomerization. Next one, after that, and again phosphorylation takes place. And second reaction is isomerization. Third reaction is like a first one, like a first reaction. What is the first reaction? Phosphorylation. In phosphorylation, what happened? ATP triphosphate, one phosphate utilized by glucose, as a result formation as glucose 6 phosphate, because in this phosphate added to the sixth carbon. 1, 2, 3, 4, 5, 6, sixth carbon. That's why it's called as a 6 phosphate. And the same isomerized called as fructose 6 phosphate. And second, phosphorylation, what happened? The same ATP adenosine triphosphate again convert into diphosphate. Where is another phosphate? It is utilized by fructose 6 phosphate, utilized by first carbon of the fructose. So by utilizing first carbon, it is converted into already fructose 6 phosphate and add another one, that is the first carbon. That's why it's called as first fructose 1, 6 diphosphate or bisphosphate. Di or bisphosphate. Because how many phosphates here? How many phosphate? Hello? In fructose, how many phosphate bonds? Hello?
Uh, one. Mm,
How many phosphate here? In fructose 1,6 biphosphate. The name itself given bi means
yes, sir. Two.
Two.
two. One phosphate at the first carbon. Another one is sixth carbon. Okay? Right? Is it clear?
Yes, sir.
Right. Next reaction, that is the fourth one, aldolose. Cleavage, actually, sorry. The fourth reaction is cleavage. Cleavage means the fructose 1,6 by bisphosphate or diphosphate cleaved into dihydroxy acetone phosphate. Dihydroxy acetone phosphate, the simply called as DHAP. DHAP, dihydroxy acetone phosphate. And another one is glyceraldehyde 3 phosphate. Is converted into two glyceraldehyde 3 phosphate. Actually, here glyceraldehyde 3 phosphate, right? Actually, the glucose carbon number, hello? Glucose carbon, hello? Glucose carbon, number of carbon in glucose. Am I audible?
Yes, sir.
Glucose carbons,
6, sir.
6, right? 6 carbon here. What about the glucose 6 phosphate carbon? Same 6 carbon. And fructose, same formula, 6 carbon. Fructose 6 also same. But in glyceraldehyde 3, after this 6 carbon compound, that is fructose 6,1,6 bisphosphate, after cleaved into glyceraldehyde 3 phosphate and dihydroxyacetone phosphate, these are the both 3 carbon. This is 3 carbon, and here also DHEAP, dihydroxyacetone phosphate also 3 carbon. And this is cleaved, carried out by aldolase enzyme. Carried out by aldolase enzyme, the fifth one, sorry, fourth one. And next, fifth one, again isomerization. That means isomerization means actually what happened here, after cleaved into glyceraldehyde 3 phosphate DHEAP, glyceraldehyde 3 phosphate directly enter into the further steps, further reaction. But dihydroxyacetone phosphate not enter into further reaction, but it is after isomerized as a glyceraldehyde 3 phosphate like a fructose glucose. After DHEAP convert into glyceraldehyde 3 phosphate, then only enter into the continuation the further reactions, right? So then fifth reaction what happened? DHEAP dihydroxyacetone phosphate convert into are isomerized into glyceraldehyde 3 phosphate, and this is carried out by isomerase enzyme. Here isomerase, this is isomerase enzyme. So in the presence of isomerase enzyme, glyceraldehyde 3 phosphate form. So anyway, first we discuss about plus first glyceraldehyde 3 phosphate. That means this one, right? This is the first glyceraldehyde 3 phosphate, and this glyceraldehyde 3 phosphate further reaction. These are the further reaction. The same reaction repeated another time. Why it is repeated another time? Hello? This this reaction, the right side reaction from the glyceraldehyde 3 phosphate to pyruvate formation, repeated it to once again. Why? Why it is repeated? Hello? Respond. All I told you, DHEAP not involved in further reaction. This DHEAP, after isomerization, again convert into glyceraldehyde 3 phosphate, which means again glyceraldehyde 3 phosphate formed. That means it would be continued until formation of pyruvate. First, already after cleavage, glycerol 3 phosphate formed, and further continuation process formed pyruvate. That is first. Second, DHEAP again convert into DHEAP, here DHEAP again convert into glyceraldehyde 3 phosphate. That is the second time. And again the second reaction would be continued until formation pyruvate, right? Hello?
Yes, sir.
Right. First, let's discuss in first glyceraldehyde 3 phosphate after cleavage. Here the glyceraldehyde 3 phosphate by oxidation process formed into 1,3 base phosphoglycerate. During oxidation process, here what happened? Hydrogen removed from the glyceraldehyde 3 phosphate. Glyceraldehyde 3 phosphate, hydrogen removed. Add to the NAD. As a result, convert into NADH2. Convert into NADH2. And as the removal of hydrogen from the glycerol 3 phosphate, here the enzyme is dehydrogenase. D means remove. Which one remove a hydrogen? So that is this enzyme name is dehydrogenase. D means removal. What removal here? Hydrogen. So enzyme name always end with ase. So it is called as dehydrogenase. And this process called as oxidation. Removal of hydrogen. Oxidation. And finally convert into 1,3 bisphosphoglycerate formed as a 1,3-bisphosphoglycerate. After that, dephosphorylation takes place. Dephosphorylation. In dephosphorylation means from the 1,3-bisphosphoglycerate, one phosphate released, removed, and add to the diphosphate ADP. Di means 2, tri means 3. Di utilizing one phosphate, it is 2 plus 1. How much? Hello?
3, sir.
Tri means
3.
3. So it is converted to ATP. And the enzyme is kinase. Same. Always the phosphate involvement kinase enzyme. So here also by kinase enzyme. Right? So, but it is not a here phosphorylation. Here phosphorylation, but here the dephosphorylation. But mean removal of the phosphate. But here add of the phosphate. So that is the difference between phosphorylation and dephosphorylation. So in first reaction phosphorylation, uh, and third reaction also phosphorylation. Fourth one isomerization. Sorry, fourth one cleavage. Fifth one isomerization. And here the sixth one oxidation. Sixth one oxidation. And here seventh one dephosphorylation. 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 What happened during dephosphorylation? Remember the phosphate removal. This removed phosphate added to the diphosphate, as a result formation of triphosphate. One phosphate removal here. So that way 1,3 becomes only 3-phosphoglycerate. Only 3-phosphoglycerate. The next reaction, mutation. The mutation means 3-carbon phosphate shifted to the 2-carbon phosphate. As a result, it is named as 2-phosphoglycerate. So this is done by the enzyme mutase. Enzyme mutase. So this is called mutation. And after that, enolase, with help of enolase enzyme, phosphoenopyruvate formed. Phosphoenopyruvate. Phosphoenopyruvate formed. Later, so last dephosphorylation takes place again. This enolase, the mutation 8th step. Enolase 9th step. Last 10th one, 10th step, 10th one. Like a seventh, dephosphorylation. So dephosphorylation, phosphoenol pyruvate, get the phospho removed, the phospho removed phospho gain to the ATP, diphosphate convert into ATP. As phospho removal, it is only remain pyruvate, or pyruvic acid form. So uh, this is one pyruvate. This is one pyruvate, right? The same mechanism takes repeated once again. So that's why another phosphate also formed. Another pyruvate also formed. So the carbon number of pyruvate, hello? Carbon number. So from the glycerol 3-phosphate until formation pyruvate, only 3 carbon. Three carbon. Then how many pyruvic acid formed? Through glycolysis, how many pyruvic acid formed? Hello?
3.
No, the carbon number 3. I am asking the how many compound of pyruvate?
1. I think,
Two,
sir.
two pyruvate formed. One is direct glycerol 3-phosphate, one pyruvate. Another one is after DHEAP convert into glycerol 3-phosphate. Again, the same reaction takes place. As a result, formation of pyruvate. So 2 pyruvate are formed. Is it clear first? Any doubt in this? Hello?
No, sir. Sir, this glycolysis is there in the respiration chapter. It's very lengthy, sir. So if we follow this pie chart
Just,
and we write
just
in the...
yes, that's enough. Only follow this flow chart and elaborate it.
Okay, sir.
Okay? The rest of the
Okay,
matter, no need. Just focus on and final end with actually you tell me how many how many NADH2 formed, how many ATP formed in glycolysis? See the chart, see the flow chart, and you tell me.
1, 2, 4 ATP, sir.
4 ATP, okay. How many NADH2?
NADH2. Only one, sir.
So actually the sum up of the for each molecule of glucose consumed in glycolysis, 2 molecule of pyruvic acid formed. That is clear.
Yes, sir.
And 2 molecule of NADH plus H plus are formed. Here in sixth reaction, here you can see here NADH2. So in first cycle, 1 NADH2, but you always consider the DHEAP reaction also. One is glycerol 3-phosphate reaction, 1 NADH2, and DHEAP reaction, another one. So here the 2 NADH2. And next, two molecules of ATP are consumed, and 4 molecules of ATP regenerated. So the net gain of molecules ATP, 2 only. That means simple. For example, okay, leave it here. How many ATP here? How many ATP formed? In
1,
7th, 1 ATP. Here, how many?
1 ATP.
1 ATP. Actually, it is repeated into 2 times, right?
Yes, sir.
One is glycerol 3-phosphate, one is DHEAP. Then how many? How many ATP? In first cycle, 2 ATP. In second cycle,
In first cycle, 2 ATP, and in second, 2 ATP. So, 2,
total 4. Right. Actually, 4 ATP formed,
4, sir.
but
2 is
the
consuming and 2 is again reducing.
right out of 4 in first reaction and third reaction. What happened first reaction? Here, are you follow marker? Hello? Is it visible to you?
Yes, sir.
Here, 1 ATP
Yes,
utilized
sir.
in first reaction. Okay. And third reaction also, 1 ATP utilized. So out of 4, 2 ATP utilized. Right? Then what is the net
Yes, sir.
amount of ATP? Net amount, only 2 ATPs. 2
2.
remember it. Remember. So 2 NADH2 and 2 ATPs are formed. Is it clear? Net gain of
Yes,
2
sir.
molecules of ATP. Right. Just wait 1 minute. Right. Next one. So any doubt in glycolysis cycle?
No, sir.
Actually, as you said, in our book, given in a reactions, fructose 1,6-bisphosphate, something reactions. You don't focus on the reaction, just write the name. First,
Okay.
first write down this. First, draw the flow chart. After that, 10 steps. 10 steps. Already explained 10 steps. You have
Yes, sir.
to give
Glucose
10
1.
points enough. 10 points enough. First point phosphorylation, what happened? Second isomerization, what happened? Third phosphorylation, what happened? That's enough. Just 10 steps enough. Okay? Is it clear?
Okay, sir. Yes,
Right.
sir.
Right. Next one. So what is the end product of the glycolysis?
Pyruvic acid. Pyruvate.
Pyruvate. Pyruvate, right.
2 pyruvate will be formed.
2 pyruvate formed. Is it clear? So, right. Now, are you see this Krebs cycle chart slide?
Yes, sir.
Krebs cycle clear? Right. So now we start with link reaction. That means end product of pyruvate, right? Hello?
Yes.
Right.
Yes,
So
sir.
this is pyruvate already formed in glycolysis as an end product. Pyruvate formula 1, 2, 3, C, 3 carbons, right? And this pyruvate convert into the C page number. This page is so page number 9. Do you have book?
Yes, sir.
The page number 92 clearly given oxidative decarboxylation of pyruvic acid. Oxidative 7.3. Oxidative decarboxylation of pyruvic acid. This is the connecting link between the glycolysis and Krebs. Glycolysis and Krebs. In these steps, what happened? After formation, this pyruvic acid, this pyruvic acid combined with, sorry, pyruvic acid combined with acetyl coenzyme A, coenzyme A, where it is given acetyl complex. Right. Simple. This pyruvate, 3 carbon pyruvate, first decarboxylation, that means remove carbon dioxide from the pyruvate, and oxidation takes place as a result formation of NADH2. That means hydrogen also removed from the pyruvate and add to the NAD, NAD, and this NAD convert into NADH, NADH2 actually, NADH2, right? And add coenzyme A. 3 steps. As a result, formation of acetyl coenzyme A. Simple. In connecting link in link reaction, what happened? Pyruvate, 3 carbon pyruvate, 3 carbon pyruvate, remove carbon dioxide, that is called decarboxylation. NAD, second, NAD convert into NADH2, NADH2, and third, add coenzyme A. As a result, formation of acetyl CoA. Now, how many carbons in acetyl CoA? Hello? Acetyl coenzyme A? How many carbon in pyruvic acid? Hello? How many carbon in pyruvic acid? Mariam,
Yes, sir.
how many carbons in pyruvic acid? Here, how many
Uh,
carbon?
Three.
Pyruvate, 3
Three,
carbon.
sir.
So after reaction, how many carbon removed?
Wa-
1 carbon removed in the form of carbon dioxide, right?
Yes, sir.
Then how many carbons in acetyl CoA?
One carbon.
What is this? From 3 carbon, 1 carbon removed. Then how many remaining?
Two, sir.
2
Carbons
only.
remaining.
2 carbons. So acetyl coenzyme A 2 carbon product. This 2 carbon product react with 4 carbon, 4 carbon, 4 carbon axial state. 4 carbon axial state. 4 carbon axial state. As a result, formation of citrate, citrate, which is 6 carbon. the 4 coenzyme, acetyl coenzyme 2 carbon as result formation of 6 carbon. And the name, the Krebs, according to scientist name, the name give as a Krebs cycle. Krebs is the scientist. He got a Nobel Prize also. And another one is after formation of that means after formation of citrate or citric acid, 6 carbon, again it is converted into isocitrate. It the enzyme involved aconitase. Remember these enzymes. Here actually enzyme not given. The remember the enzyme name is in coenzyme A to form as a citrase. Here the enzyme is synthase. Enzyme synthase. Please remember the enzyme names. Synthase enzyme. Synthase enzyme. Citrate convert into isocitrate in the form of aconitase. Right? This isocitrate went to the oxidation process as a result formation of alpha ketoglutarate. Remember this intermediate product. Right? And first of all, citric synthetase clear, isocitrate clear. Actually, before that, cis aconitic acid form. After that, isocitrate. Okay, remove it. Isocitric acid. And after formation, isocitric acid, this isocitric acid with help of dehydrogenase enzyme and isocitric acid. Right. Dehydrogenase enzyme. That means alpha ketoglutarate enzyme formed into alpha glutarate. Alpha glutarate. Isocitrate finally convert into alpha ketoglutarate. Before formation alpha ketoglutarate, the one carbon molecule removed. Decarboxylation also takes place. Isocitrate oxidation takes place. In oxidation, what happens? NAD convert into NADH2 1. And after that, decarboxylation takes place as a result formation of alpha ketoglutarate. Then how many carbons in alpha ketoglutarate? Hello? This is the first oxidation process, remember. First oxidation process. How many? How many in
Four.
alpha ketoglutarate? How it is 4? How many in isocitrate? How many in citrate? How many carbon in citrate? Hello? Citric acid,
5,
how many carbon?
6.
6 carbon. Isocitrate also 6. There is no removal of carbon dioxide. But isocitrate, when convert into alpha ketoglutarate, the two reaction takes place. Oxidation and decarboxylation. As the decarboxylation takes place, one carbon loss. As a result, the number of carbons in alpha ketoglutarate, how many? 5 carbons only.
5.
Right, remember it. After that, second oxidation takes place from alpha ketoglutarate, NAD convert into NADH. This is the second oxidation process. As a result, formation of succinyl coenzyme A, succinyl coenzyme A. After formation succinyl coenzyme A, here GTP formed. GDP, utilized phosphate, formed into GT. Or ADP convert into ATP. Remember, in animal GTP form, in plants ATP form. Okay? Hello?
Yes, sir. Sir, full form of GTP is?
Guanine triphosphate.
Okay.
ATP adenosine tripose. Actually, here the name not clear. Anyway, succinic acid after formation, succinyl coenzyme A, succinyl coenzyme A, succinyl coenzyme A, fumaric acid, right? Something missing here. Just wait. This is succinic acid. Remember this word. Actually, this is not clear. This is this one, succinic acid. Succinyl coenzyme A utilizing phosphate and as a result convert into ATP, JADP convert into ATP and as a result formation of succinic acid. Right after formation, this is succinic acid, remember it. After succinic acid, another oxidation process takes place. This is the third oxidation process. In third oxidation process, what happened? FADH2 convert in FAD convert into FADH2. FADH2.
Sir, what is FAD?
Flavonine adenosine dihydroxate. NADH2 nicotinamide. FA flavonine. And after oxidation, fumaric acid formed. Here also cut the name. This is fumaric acid. After formation, fumaric. If you see in book, fumaric acid. From fumaric acid, add water molecule and form malate or malic acid. Malic acid. And again here the fourth oxidation takes place. In fourth oxidation, what happened? In fourth oxidation, NAD convert into NADH2. As a result, formation of oxalo acetate. Here, before formation, malate, here the removal of carbon dioxide also, decarboxylation takes place. Decarboxylation takes place from the, after malate, during malate, convert into oxalo acetate, carbon dioxide also removed. Decarboxylation also takes place. I think it is not clear. Malic acid. How many carbons in malic acid? Just wait 1 minute. NADH2, okay. Fumaric acid, okay, clear. Malic acid, okay, clear. Then where the decarboxylation takes place here? Where? Just wait 1 minute. Succinic, fumaric, malic acid, okay. Malic acid, oxaloacetic acid. Where is the fumaric? Fumaric, malic acid, okay. Malic acid, okay. So, okay, something miss here. Just remember, during malate to convert into oxal acetate, get the carbon dioxide removed, right? So another carbon dioxide removed to formation oxal acetate. Before formation oxal acetate, how many carbons are there from alpha ketoglutarate to the malate? How many carbons? Hello? How many carbon in alpha ketoglutarate? Here, how many carbons?
Five,
Five
sir.
carbons. And here another one carbon removed. Then how many carbon in axial state?
4, sir.
4 carbon remain. So we start with 4 carbon axial state. the cycle, the Krebs cycle start with 4 carbon oxyl state. Again end with 4 carbon oxyl state. So these 4 carbon oxyl state react with 2 carbon acyl coenzyme A as a result formation of 6th carbon citric acid, right? Then
Yes, sir.
you tell me how many NADH2 are formed. How many NADH2 are formed? Listen carefully. How many NADH2 formed in Krebs cycle? Already given number also. Number of oxidation. How many number of oxidation here? Oxidation number. This is first one.
3,
First
sir.
oxidation. First, right. 3. First oxidation, NAD. Second oxidation, NAD. Here the third oxidation. This is third oxidation, but it is not a NAD. What is here? FAD, convert into FADH2. See this, see in book also. If the slide not clear, if the slide is not clear, see in book. Third oxidation, FADH2. And fourth oxidation, again NAD convert into NADH2. Right? So in this process, as you said, 1, 2, last one, 3 NADH2 formed. Right? But
Yes, sir.
this Krebs cycle, how many times repeated? Krebs cycle, how many times means how many pyruvate acid formed? How many pyruvates are formed after glycolysis?
2, sir.
2. That's why the Krebs cycle also turns into 2 cycle. So first
Okay.
cycle 3 NADH2, right?
Yes, sir.
In second cycle, again same cycle. That means
3.
3. Total 6. 6 NADH2. NADH2 formed in Krebs cycle. 6 NADH2 formed in Krebs cycle. Then how many FADH2 are formed? How many FADH2?
Mm.
How
1 only,
many?
sir.
1 for 1 cycle.
2.
You always remember this cycle turns into 2.
Yes, sir. 2.
Then how many FADH2?
2 only, sir. For
2
one
only,
cycle,
right?
1.
Right. So 2 FADH2. 2 FADH2. Then how many ATPs are formed? How,
ATP.
how many ATPs here?
1 only, sir.
1. But this is for 1
So
cycle.
2.
Right. 2 ATP. So remember in Krebs cycle, 6 NADH2 form, 2 FADH2 form, 2 ATP are formed. Is it clear?
Yes, sir.
This number important. This number important for balancing sheet to the electron transfer system next cycle, next class. Right? So in glycolysis, 2 NADH2, 2 ATP. In Krebs cycle, 6 NADH2, 2 FADH2, 2 ATP. Is it clear?
Yes, sir.
Any doubt in this?
No, sir.
Any doubt? Glycolysis, Krebs?
No, sir. For Krebs cycle also, if we follow this flow chart, that is enough, sir.
Enough. Glycolysis and also Krebs, both are just to follow the Krebs cycle and write the content, what you draw, what is the graphical representation given in Krebs cycle or glycolysis. That much enough. You write. No need to extra matter. No need to write extra. Only
Okay.
first to draw the flowchart and explain the flowchart in your own way. Okay?
Yes, sir. Sir, in this cycle, GTP is also there, no? So that also we have to count.
No. Only ATP. Only ATP.
Okay.
GTP for animal. So only ATP.
Okay.
Okay, clear?
Yes, sir.
Right.
Thank you, sir.
Right.
Thank you.